Hey everybody, this is a video version of how to do the assignment. The solution I'll post online as a PDF file or a JPEG file, but I figured this video might help you guys if you guys are stuck on a question and you're just wondering how to go about solving it. So the first thing we'll do is we'll start with number 1, obviously. And the question asks, in a certain organism, N equals 6. N is the haploid number. That means that diploid means 2N. Thus, if N equals 6, then 2N must be 12. So the answer is C, 12. For number 2, a chromosome contains 2 sister chromatids joined by a centromere. And if we observe the cell before meiosis 1, we know that the chromosome contains 2 sister chromatids connected by a centromere, and that there are 12 of these. And what this means is 2N equals 12 means that there are 12 homologous chromosomes present. So when the homologous chromosomes line up into their associative pairs, and since we know that each chromosome at this point contains 2 sister chromatids, it means that there are 24 chromatids in total in this cell. Now, once it divides in meiosis 1, we know that the homologous pairs split. They're no longer a tetrad, and each homologous chromosome goes into a separate cell. So in this first cell we have 12 homologous chromosomes, but in each of those chromosomes we have 2 chromatids, meaning there's 24 chromatids. Okay? Please be aware of the difference between chromosome and chromatid. Meaning that in meiosis 1, we have 12 chromatids in each. Because we're going to have 6 of those homologous chromosomes in each cell. So we have 12 chromatids. Alright, so now number 3. Which of the following statements applies to homologous chromosomes? Well, it's not A, because sister chromatids, they're the ones attached to the centromere, not the homologous chromosomes. When homologous chromosomes pair up, they form a tetrad, but they're not attached by the centromere. They cross over, but not at the centromere. It's not B either, because they're not genetically identical. Homologous chromosomes, they always line up as pairs, right? And in each of those pairs, one homologous chromosome belongs to a mother, one homologous chromosome belongs to a father. So let's say we have these homologous chromosomes coming together to form a tetrad. One homologous chromosome of the pair belongs to a mother, the other homologous chromosome of the pair belongs to a father. So they're not genetically identical. It's not C either, because there aren't any homologous chromosomes in cells that conduct mitosis. Because there's no need for them to come together as a tetrad to genetically exchange information. It is D, because one homologous chromosome may contain a variation of a gene which we call an allele, and that's going to be different than the other. So these alleles, when they come together, these homologous chromosomes, when they come together, they exchange alleles. So the genes are all in the same order, it's just that each, say one gene of one chromosome is going to be different from the other, and that's what we refer to as alleles. So the answer is D. Moving on to number 4, which of the following events occurs during meiosis but not mitosis? Pairing of homologous chromosomes only happens in meiosis. because the pairing of the homologous chromosomes creates tetrads, and those tetrads undergo crossing over, genetic exchange. This does not occur in mitosis, only in meiosis. So the answer is B. For number 5, we're going to first answer 5B, just to show your work, just to demonstrate how you go about doing it. So, non-disjunction in meiosis 1 causes 2 cells to be missing a chromatid. So, as you know, here's our homologous pair. Non-disjunction in meiosis 1 causes both of these to not divide correctly, meaning that they both end up in one cell but not the other. So if we were to continue that, we'd see 2 cells with 1 more chromatid than they should, and 2 cells with 1 more chromatid missing. Be aware that these aren't actually empty. We're just referring to this specific chromosome. So say if it was chromosome 1, we're just talking about chromosome 1 here. They're not actually empty. All the other chromosomes we assume are there. But the point I'm trying to make is that non-disjunction in meiosis 1 causes these chromosomes to be placed in this cell, which causes ultimately the resulting cells to have 1 more chromatid than they should. So this is, this is under the assumption that meiosis 2 conducts properly. Now, with that said, for a mother, that would mean that the egg could possibly be missing an X chromosome. Thus, when a sperm with an X chromosome comes and fertilizes this egg, or this egg, that's when we'd result in an XO. Say if it was a reverse, say if this was a male, and we have these sperm cells missing an X chromosome, and it heads towards an egg with an X chromosome. This sperm travels, fertilizes this egg, we'd also end up with an XO, because the female is providing the X, but the male is missing the O. So it could happen to the mother or the father. This is what would occur if non-disjunction occurred in meiosis 1. In meiosis, if it were to only occur in meiosis 2, we know that, let's see here, I'm just going to draw it out. So these cells divide properly in meiosis 1. This goes properly in meiosis 2. But say this one does it improperly. We have one cell that has an extra chromatid. And this would play out, and another cell that doesn't have a chromatid, is missing that chromatid. So this scenario could play out just as it did here, where we have a sperm fertilizing an egg that's empty. Same with this one. Say this was a sperm, it could fertilize an egg with an X, but because that sperm is empty, it results in an XO. Or the other way around, where this is the egg, this is the sperm cell, and this fertilizes that, and that also results in an XO. So to answer the question in 5A, in humans, which of the following events could not give rise to an XO female? It could happen to any one of these, meaning that all of the above could give rise to an XO female. Alright, non-disjunction could occur in the first or second division of meiosis. XYY individuals would most likely arise from the non-disjunction at something, meiotic division in the something. So what we're looking for here is the XYY individual. We have to, again, look at non-disjunction. And for a cell to contain a Y chromosome, it must regard the father. So we know that the YY individual is what we're looking for. We know that the father is involved. So, we can discount the mother. The mother provides no Y chromosomes at all. For a cell to contain 2 Y chromosomes, non-disjunction must occur at the second meiotic division. So just going back to up here, what we drew up here. Say this was talking about our Y chromosomes. To have an extra Y chromosome, it must occur in the second meiotic division. This would result in, say these were Y chromosomes, 2 Y chromosomes that would fertilize an egg that has an X. This would result in an XYY. Meaning that it is the non-disjunction at the second in the father. So the answer is D. Now there is a scenario where you have an X chromosome and a Y chromosome, and non-disjunction happens here. Right? In meiosis 1. And then there's also that possibility where non-disjunction also occurs in meiosis 2 here, where we have an X chromatids, 2 X chromatids here, and 2 Y chromatids here. That's also possible, but that's just saying that non-disjunction occurs in meiosis 1 and meiosis 2. And then in the context of our question, it's only giving you the option of, first or second, not first and second. In this scenario, it's first and second. Just a side note, just to be aware of that in case you came across that sort of conclusion.